All right, hey, seventh graders. So today we're going to start on protein synthesis. And so remember, anytime you hear the word synthesis, that's the same thing as to make. Okay, so we're going to make proteins today. And so if you took away anything from, let's see, you're going to have this on Friday, Wednesday. So Wednesday, you guys made a DNA molecule, a short little section, right? You made 4 base pairs. Adenine, you paired up adenine with thymine, and then thymine with adenine, and then guanine with cytosine, and then cytosine with guanine, right? And you made a 4 base pair ladder, right? And it was 2-sided to it. So then you guys hopefully started taping all of yours together as a class, and then you guys should have a long DNA strand, right? And then this is what we all have inside of our DNA, inside of our bodies as well. And all of these DNA sequences can be very different. And they have to be different in order to make different proteins, okay? So remember, DNA is this instructional molecule in our cells that tell our cells how to do everything, how to make all of these molecules, how to make structural molecules, like we have wood in our walls, 2x4s that hold up our houses, right? That's structural, that holds up our houses. Or they can make more functional things like proteins. And that's what we're really going to start talking about today is proteins. And now these things that perform functions, that go out and do things, right? That go to our stomach and break down our food in our stomach so that we can get nutrients, or that begin to make things like our hair, make our eye color and things like that. These proteins do all of these jobs. They're super important. So when you generally think about proteins, you might think about like a nutrition label. You're like, okay, this food has a lot of proteins, going to make me big and strong, which that's absolutely true. But proteins do a lot more than that. Okay. So let's go ahead and get into this. Right now, if you guys want to go ahead and pause this, we're going to go ahead and go through this DNA and RNA transcription translation notes. And so your notes packet should look like, or packet, it's front and back, right? So it starts with transcription here. It's filling the notes, looks like this, goes into translation, and then here's the last page. It ends with codons. So this is what you should be filling out with this PowerPoint. This first part, I will tell you when to start taking notes. This first part is talking about replication. And we're just going to talk about it real fast. So DNA obviously has to replicate, right? We have cells that die and we have more cells that we make. And so in order for that to go on, there needs to be DNA in these new cells. And so we have to replicate our DNA. And so DNA is copied by itself, right? Because when new cells are made, the cells have to have an exact copy of that DNA. So if I want another skin cell that does the same function as my old skin cell, then that means the DNA within that skin cell has to be exactly the same, right? If you want a cell to do exactly the same thing as its parent cell did, then you have to program it the exact same way. This occurs in the cell cycle. So during mitosis, during meiosis, where we talked about these cells replicating, during that short S phase, that synthesis phase, is where DNA is replicated. This process is called replication. It's considered semi-conservative. Why is it called semi-conservative? Because we're actually only making one side to our DNA. Similar to what we did on Wednesdays, I already told you guys, all right, here, put adenine, thymine, guanine, and cytosine and make those nucleotides. And then I'm going to have you make the 4 complementary nucleotides that go with those guys, right? So if I said make adenine, well, what pairs with adenine? We know adenine always pairs with thymine, right? And then I said, all right, here's thymine. What always pairs with thymine? Adenine. So then you went ahead and made adenine. Well, all right, I told you to put on cytosine. Now what pairs with cytosine? Guanine. So you made guanine. And then I said, all right, you have guanine over here, and now you need to pair that with cytosine. Okay, so semi-conservative means one side's already made, where then your body reads that side and then makes the other half of the DNA. So it doesn't make the entire double strand. It only makes one side of it. So replication occurs, DNA is untwisted. So we know that DNA is a double helix. The old DNA strand is a template, and so it's just like something to copy off of. So anyways, we can start seeing this little process here. Like I said, you don't need to be taking notes yet. So we have our double-stranded DNA. Our DNA is being split in two. Whoa. And so here's the template. So here, the blue strand up top here is the old one. And now this DNA polymerase 3 here is creating the new strand of DNA. So this will become a new strand of DNA. This strand is also the old one, but it's also going to create another new strand of DNA. But only one strand is being created, right? So what I'm trying to say here is both strands are not being thrown away and then a new double-stranded DNA molecule is being created. No. The original DNA strand is being split into two, and then a new half is being built on to each of those originals. All right, so we've seen this picture a million times. We have this DNA here that's wound up, and we can wind it up super duper tight into a chromosome. All right, and so this is just another picture of how DNA is being replicated. DNA is not all replicated. It doesn't start at the very beginning of the DNA strand and then it replicates the entire thing. That's not how it works. It actually starts at different points along the DNA. That's not super important to you guys, so we'll just keep on moving. There is then, so we talked about mutations on Wednesday. So when DNA's, anytime, like if you are copying something, there's always a chance that you're going to mess up, right? If you're copying somebody's writing, you may misspell something or you may switch up some words, right? That's a mutation. You change something. Now the meaning is different. So DNA actually has these molecules called enzymes, and they are proteins that are made from the DNA. They come along and they read the DNA and they're a proofreader. And they make sure that everything is correct. Because what's important? Well, if we have a skin cell that dies, we need to replace that skin cell. That skin cell needs to have the same exact DNA that its parent before it did, right? So these proofreaders come along and make sure that DNA is perfect. And they do a really, really, really good job. They read millions of base pairs in minutes, right? And if you could do that, you'd be superhuman. So if mistakes are not fixed, these guys can cause problems, and these are known as mutations. So some of these mistakes do get past these proofreading enzymes, but proofreading enzymes, they are very efficient, and it's been proven that they do a very good job. Again, something you can know, but we don't have to write this down. All right, so we have our parent molecule up here. Double-stranded, right? Adenine always pairs with thymine, cytosine with guanine, thymine with adenine, adenine with thymine, guanine with cytosine, right? This parent strand in replication, it gets split in two, and now each parent strand now serves as a template. So here's our template. So this is the original, and now a new DNA strand is being placed on it to, replicate the DNA. And so this old DNA strand determines the order of nucleotides along the new complementary strand. So if I look at these light blue guys here, T, G, A, T, C, right? They're the same exact as a T, G, A, T, C over here, right? And that makes sense because we know adenine only pairs with thymine, cytosine only pairs with guanine, and vice versa. Okay. You guys don't have this, but I do have this one for transcription. Okay, so the central dogma. Central dogma means what's the main idea behind this? And so we just got done talking about traits and genetics, right? Heredity is the study of how traits are passed from parent to offspring. And what are these traits? How are traits shown, right? How are brown hair traits, blonde hair traits, blue eye traits, green eye traits? And we talked about height, right? We talked about skin color. There's something called lactose intolerant. If you're lactose intolerant, that means you can't drink milk because there's a molecule called there's a sugar called lactose in milk. And that means you don't have the enzyme, the protein called lactase, to break down that sugar. So that if you're lactose intolerant, you get upset stomachs from drinking milk because you're unable to break down the stuff in that milk. And that's because you don't have the proper gene to make the molecule that breaks down the sugar in milk, making you lactose intolerant. Okay, so that was a little bit of a segue, sorry. The central dogma states that information flows in one direction from DNA to RNA to proteins, right? So if we have the nucleus, right, we have all of our DNA in the nucleus. We've always known this. DNA is always replicated in here. And then we're going to talk about a new molecule. So DNA gets transcribed. So you see this process called transcription. It happens in the nucleus. We'll talk about it. DNA is transcribed into RNA, so it's kind of turned into a different language. So it's kind of like somebody in China is trying to talk to you and tell you how to do something. Well, you need somebody to transcribe what they said in Japanese, and they need, or I don't know if I said Chinese, so maybe Mandarin. I can't remember what I said. Anyways, so now they're going to transcribe what their language to English so that you can now understand that and carry out the orders that they wanted you to. So DNA is transcribed into RNA, and then RNA is translated into a protein, meaning it's going to make the proteins that express these traits that we've been talking about for the last month or two. All right, so first things first is transcription. Here's where the notes start, so go ahead and get ready. All right, so transcription is the process of mRNA copying DNA. So that M, so DNA stands for deoxyribonucleic acid, and we've talked about that a few times. RNA is very similar, except RNA stands for ribonucleic acid. So you're just missing the deoxy. So it's just ribo. Where's my cursor at? Ribonucleic acid, right? And the M in front of it stands for messenger. So this is a messenger molecule. So messenger RNA is copying the DNA. This is how new mRNA is made. It copies a gene. So again, proteins are made from a single gene, and we have over 25,000 genes in our DNA, right? And so what will happen is only one protein is made at a time. And so that mRNA will go to that gene, and it will read that entire gene and will copy itself a piece of mRNA. This process occurs in the nucleus. This occurs just like replication, but with RNA polymerase instead. So what's that mean? Polymerase, anytime you see ASE, so we see polymerase, or like I said earlier about lactase, if you see ASE at the end, that means it's an enzyme. It's a protein that does a job. So lactose is a sugar in milk, and lactase is the enzyme that breaks down lactose. So that's kind of helpful, right? So only one side of the DNA is copied, and then the nucleotide. So we know that DNA is composed of 4 different nucleotides, right? We know that we have adenine, thymine, guanine, and cytosine. However, when we in the process of transcription here, thymine is no longer with us. Thymine gets kicked out and is replaced by another nucleotide that we call uracil. So right here, nucleotide thymine is replaced by uracil during transcription. So when we look at our single strand of mRNA, we won't actually see any T's or any thymines on there. Instead, we'll see uracils. Okay, so DNA has thymine, uracil, and then mRNA has uracil. So there's really 2 big differences between DNA and RNA. So DNA is double-stranded, has thymine. RNA is single-stranded, and it has uracil instead of thymine. So 2 big differences right there. All right, and then what you guys can do, you can pause this, and obviously if I'm going too fast, if you guys can't keep up, I'll take notes, pause this video at any time. So anyways, here's where you guys can practice this, and down here, go ahead and write what would pair up here. So you're making mRNA here. You're not making another DNA strand, you're making mRNA, okay? So this is single-stranded. So what pairs with thymine? Adenine. What pairs with thymine? Adenine. So I'd put A. What pairs with guanine? Cytosine. C. What pairs with cytosine? Guanine. G. What pairs with thymine? Adenine. What pairs with adenine? Thymine. Wait, not thymine, because we're making mRNA. So now here's where you would put your uracil, the U. And then G, what pairs with G is that cytosine. And you guys can go on and do the rest of these. I'll do them real fast again if you want to pause this so everybody can do this practice real fast, because this is, the activity is going to be based off of this stuff. So you have to make sure that you know how to do this. So this is transcription only. This is not DNA replication. This is transcription. We are going from DNA molecule to a messenger RNA molecule that will then turn into a protein outside the nucleus. Okay. So next, thymine pairs with adenine. Adenine, again, pairs with uracil because we're making mRNA. Guanine pairs with cytosine. Cytosine pairs with guanine. Guanine pairs with cytosine. C, G, T, A. A, U, G, G, C, A, G, and then U. And then this last one would be C, G, A, U, uracil, A, C, A. Okay, so that's where your practice should be at. If you need to stop that, slow it down, and write it on the board so everybody made sure they got it right, go ahead and do that. So here's kind of the process, and I have a short little video to show you guys on this whole thing, because I know this is a very new subject, so we'll make sure we get this idea down. So here's our DNA. So right here it says RNA polymerase. So again, polymerase, if it has ASE at the end, that means that it's an enzyme. It does some job inside the cell. So we have this big globular molecule that attaches to your DNA. It finds the gene that it wants to express, right? Let's say you need blue eyes. All right, so this RNA polymerase rides down the DNA, and then it finds the gene for blue eyes, and then it goes, okay, I found it. And now it starts making this messenger molecule, messenger RNA, for blue eyes. And so that's what this, so down here it says newly made RNA, this pink strand here. And so it starts pairing, right? Here's the blue is the old DNA copy, and then the pink's the RNA. So adenine, uracil, right? Thymine's been replaced in the mRNA. C goes with G, C goes with G, A, so on and so forth. Okay. I'm going to go ahead and skip this because, like I said, I already have a video made up for you guys. All right, so that's transcription. Transcription is just going from DNA to excuse me one second, somebody's giving me a call here. I'll have to pause this. All right, and we're back. Okay. So now on to translation. So translation is taking that mRNA strand that we just made inside the nucleus. So that mRNA strand is single-stranded. It has uracil instead of thymine. And now we're taking that messenger RNA and we're taking it outside of the nucleus into the cytoplasm. And if you guys remember the ribosomes from earlier on this year, ribosomes are these little itty-bitty molecules that are speckled all throughout the cytoplasm that make proteins, okay? And so now we're really going to take a look into what those guys' jobs are. Okay. So translation. So this is also known as protein synthesis. DNA is ultimately a code for protein production. Protein, in turn, make all the parts of the cells, which will then control all of the chemical processes. Proteins are made up of amino acids, okay? So amino acids, this is going to be a new molecule that we have to learn about. So amino acids are these little molecules that we chain together, and we put a whole bunch of these guys together, we make up this protein, okay? And that's all it is. So translation, mRNA transcribes the DNA, gets the message code, so that's part of, we just called that transcription. mRNA carries that message from the nucleus to the ribosomes. It could be on the ER, could be free-floating ribosomes. And then the ribosomes reads the mRNA. And really that's where translation starts, is once the ribosomes meet the mRNA and start making the proteins. So now it says the rRNA. Now we got a little R. That just means ribosome. So our ribosomes are actually big old clumps of RNA. So rRNA is just ribosomal RNA. It's talking about a ribosome. So a ribosome tells the tRNA, oh gosh, now there's another one. T stands for transfer RNA. These transfer RNA guys, these guys bring in these amino acids from wherever they are in the cell, and then they start chaining them on and making these proteins. And like I said, I'll show you a little video that helps out with this. These are needed by reading the codons. You're like, what the heck's a codon? Well, I'll tell you. tRNA collects and brings back the amino acids. The rRNA, remember, it's just the ribosome, hooks together the amino acids into a protein. Okay, so here's the codons. Codons are every 3 nitrogen bases. So anytime we have adenine, thymine, guanine, so there's 3, or thymine, guanine, any kind of order that these guys can be in. So we see some of these AUG, so adenine, uracil, guanine. This is specifically a start codon. It's telling the ribosome, all right, start protein synthesis. This one right here, so we have a couple stop codons. And so these are 3 base pair segments that say things. And this is going to be part of the activity. So you have to really get what a codon is here. So if you get uracil, adenine, that means stop. If you get uracil, guanine, adenine, that means stop. If you get uracil, adenine, guanine, that means stop. On the tRNA is the anticodon. It's complementary. Anti means the exact opposite. Doesn't mean bad. It just means the exact opposite. So the anticodon, so if there's a uracil, adenine, adenine, that means what pairs with uracil, adenine. What pairs with adenine, uracil, and uracil. So actually, what the anticodon would look like for this stop codon would be AUU. The anticodon for this would be ACU. The one for this, the anticodon for this one, remember it's just the opposite, would be adenine, uracil, cytosine for this one. Okay, and so let's take a look into what's all this mean. I think that's it. Okay. Let me talk about this real fast. So here's a I'll go ahead and send this out to you guys. And so here's all of the possible combinations that you can have with your uracil, cytosine, adenine, guanine, right? And these codons are always 3 base pairs. And so first we need to find, okay, here's our first base pair. Is it uracil, cytosine, adenine, or guanine, right? So all of these guys here, it says U. If I go across here, all of these guys start with a U, a uracil, right? And then going down, so it says the second base. So now the second in the codons is going to be uracil, 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 uracil are the second guys. Here, if I come over here, the second base is going to be cytosine, 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 and all of these. And then if I come over here, the third one, if I come over here, uracil, 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 all of these guys have the third uracil. Cytosine's right here, third one cytosine here, third one cytosine. And then we see these phenolphthalein, leucine, leucine, isoleucine, methionine, valine, alanine, thronine. You don't have to remember all of these. They're all going to be written down for you. So don't worry about memorizing all of these guys. I never had to. There's 20 amino acids, 20 different amino acids. So some of these we see UAA, UAG, UGA says stop, stop, stop. So these are the stop codons that tell protein production to stop. And then AUG, methionine, is actually the start codon as well. Some of these double time, so GAU and GAC, both of these codons make the same kind of amino acid. Okay. So let's keep going on. You may be a little confused right now. That's totally fine. I got something else that's going to help out. Okay. So here's translation. So again, here's the single strand of mRNA that's coming out of the nucleus, and now this mRNA strand comes in contact here with the ribosome. Now we have this tRNA guy that brings the anticodon. So 1, 2, 3, the 3 base pairs that are the opposite of what's on the mRNA strand. Attached to the tRNA is an amino acid right here. So as these tRNA, remember they're transferring these anticodons, that's why it's a T. As they bring this new amino acid here, they begin to link these amino acids, then grow into a chain. And so if we see on down the line, this amino acid chain has grown even more, and down the line has grown even more. Translation occurs in the cytosol for both eukaryotic. Eukaryotic are the guys, us. We have a nucleus in our cells. Prokaryotic cells are like bacteria that do not have a nucleus. But I'm going to go ahead. So here, it shows how, all right, here's our start codon, methionine. So AUG on the mRNA. Here's the anticodon that tRNA has. So tRNA is this guy. He brought the anticodon, uracil, adenine, cytosine. Attached to that is the amino acid methionine. The next one that's coming along with the anticodon that matches with the mRNA here. So remember, it's 3 base pairs. He's bringing another amino acid. And so this is a peptide bond. Well, another word for a protein is a polypeptide. So again, can be a lot of vocabulary. So we'll get into that more next week. But then, as you can see, methionine and leucine have now been put together. Now cysteine is coming along. Now we see this growing chain of amino acids that's growing. And once it gets so big and it gets at the end of its mRNA, it says stop. And now this protein has been made. And now we'll go on and do other functions within the cell. Okay, so that should be it for your guys' notes. So let's go ahead and run on to this video here. And this might be for second and third hour, but I'll go ahead and leave this up to the sub. I'm going to go ahead and play this video, and I'm going to play it on silent, so you guys only listen to me. All right, so we have
Here is a
our
cell,
here.
The basic unit of all living tissue.
Here's our cell. We go inside of our cell.
In most human cells, there is a structure called the nucleus.
This looks like a Golgi body. Here's our mitochondria.
The nucleus contains the genome.
That we're really concerned about.
In humans, the genome is split between 23 pairs of chromosomes.
Pairs of chromosomes. Inside of these chromosomes,
each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
Histone proteins that help them coil up and help them compact.
Within the DNA are sections called genes. These genes contain the instructions for making proteins.
Right? It's just a section of the DNA that encodes for
when a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene.
So RNA polymerase,
it moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus.
All right. So this RNA polymerase, remember, if we say ASE at the end of the name, that means it's an enzyme. It's functioning. It's doing work inside the cell. So what's it doing here? It's unzipping, just like a zipper. It's unzipping the DNA strand, and now what it's doing is taking other nucleotides and it's pairing them with them, right? So what pairs with thymine? Adenine. What pairs with adenine? Not thymine, because we're making mRNA, right? So uracil has now come into play. Well, pairs with cytosine, guanine, and so on and so forth. And so now we're making this single-stranded mRNA, this messenger, right, that will eventually make its way out of the nucleus.
The DNA code determines the order in which the 3 bases are added to the messenger RNA.
Are building.
This process is called transcription.
As a template strand. So now here's this elongated, or this messenger RNA.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA.
All right, so every once in a while there has to be stuff that's snipped out.
The messenger RNA then moves out of the nucleus into the cytoplasm.
Goes out of the nucleus into the cytoplasm, and now it's going to find a ribosome, or the rRNA.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA.
On top of this mRNA.
The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome.
Will then be hooked to another amino acid.
The messenger RNA is read 3 bases at a time.
So here we go. We see this anticodon, and if these anticodons are like, oh, okay, what pairs with AGU? Well, I know that UCA pairs with this guy, so therefore I know that this amino acid that's on top of this transfer RNA here is in the right order. Right?
As each triplet is read, a transfer RNA delivers the corresponding amino acid.
This amino acid adds another amino acid. And so
this is added to a growing chain of amino acids.
Again, a polypeptide is another name for a protein. They're the same thing.
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
They'll start to fold, they'll start to form crazy shapes because the shape determines the function of what this protein does, or determines its structure as well. All right. So this is on my plan book page. If you guys want to play through it and listen to what the lady has to say, I pretty much just said what she did. All right. So let's go ahead and start with your guys' activity. So you can go ahead and pause this video, put your notes away in your folder. And so now we're going to go ahead and start this. So what you guys need to get out now, up on the board, you should have a couple sheets of this. So what these are these are genes, okay, right here. So here's gene number 1, essentially. Here's gene number 2. So see how these are already put into 3 nucleotide base pairs, and that's what we want, because this TAC, right, this is DNA, so thymine is still on this guy. TAC, if you notice, they all start with TAC. That's actually called a start codon, okay? And so your sub is going to say, put you guys into groups of 2. And when you guys get in groups of 2, then the sub's also going to say, all right, this group, you guys get gene 1, 2, and 3. And then group 2, you guys get 4, 5, and 6. And then the next group, group 3, get 8, 9, and 10. And then start over. So everybody's kind of doing different pieces of genes, right? So now what you're going to do is you're going to take this gene that's located on the DNA and you're going to turn it into mRNA, right? And so, like I said, there should be a couple copies of this up on the whiteboard, so you guys will have to get up out of your seat, and you have 3 of these guys. Okay, so here's your sheet that you should have. So you have your DNA, and now you're gonna take your sentence 1 for DNA, and it says what's the mRNA. So now you're transcribing this guy. So all I'm doing is I'm turning it into an mRNA code. So if I'm doing sentence 1 here, and I come up to here, so TAC, let me get my screen out of the way. So what pairs with thymine, adenine? What pairs with, right, I'm here, sentence 1. Thymine, what pairs with thymine, adenine? What pairs with adenine? Well, again, we're making mRNA, so it's not thymine anymore, it's uracil. And then again, what pairs with C? That is G. Okay, so I'm going to write that on the first space, right on here. And then what pairs with G? Cytosine. What pairs with G? Cytosine. What pairs with A? Thymine. All right, and you're going to do that with your 3 specific sentences, right? So like I said, group 1 will have 1, 2, and 3. Go up on the whiteboard, find these, write them down, take them back to your desk, and then transcribe them into mRNA. Okay. Now next, you're going to be looking on my big table up in the front of the room where usually instructions going on. So it might get a little crazy, or it might get a little crowded. Not crazy. You guys don't get crazy. It might get a little crowded. So just be patient. Now what you guys are going to do are find those codons. Okay. And so these codons are 2-sided, and here they are. And so if I look at these guys, I see here's a codon, UUG. Okay. So on the back of this page for UUG should be stop. So if I have the sheet in front of me and I see, what I say, AAU? No, I said UUG, sorry. All right, so I see UUG. Okay, I go to find UUG, I flip it over, it's the stop codon. So then I would find that codon back here, right? I wouldn't start with a stop codon, so then you'd mess up somewhere. But as you can see, all of these have words. Well, what you're doing here is you're going to make an English sentence. So sentence 1 here, you have these codons in your mRNA, and you're going to translate them, right? You're going to make a protein, but you're not actually making a protein. You're making a sentence, okay? And so when you make a sentence, the sentence obviously should make sense. So you have sentence 1, sentence 2, sentence 3, that you're going to sift through these. And you're going to, once I find this codon UCU, I'm going to flip it over. And what's on the other side, if it says protein, I'm going to write down protein in that same spot. And you should put the words in the same order that these codons appear in. Okay? And so it shows you here what's the amino acid that actually codes for glycine. We'll talk more about amino acids, but right now just focus on how our DNA inside of our nucleus has genes. Those genes are read and they're transcribed into messenger RNA, that mRNA. So now we have made this mRNA. That mRNA then floats its way out of the nucleus. It finds a ribosome. Now this ribosome reads these codons, UCU, or let's go with CCA, and it reads that and it says, okay, CCA, that's the amino acid leucine. So then it brings the leucine amino acid and tacks it on there and it keeps growing this chain. But what you're going to write down is protein because that's the next word in the sentence that you are making. Okay? So after you guys get done with that, make sure you guys' names are on these. And all you need to do is just have your sentences 1, 2, and 3 finished up. And you need to turn that in. This will be due for, when will I see you guys? Tuesday, and I will be back to school. Okay? Your guys' tests are up online right now. If you guys haven't seen those already, if you have any questions, please message me. Other than that, good luck.